Welcome to another screencast where this time we'll be discussing muscle tissue, and by the end of this hopefully you will be able to identify, describe 4 functions of the muscular system and then the general characteristics of the 3 types of muscle tissue, as well as then get a chance to practice looking at the histological preparations, that is microscope slides of the 3 different tissue types and being able to identify them. So let's get started. Obviously here the muscular system is first and foremost responsible for producing movement. Okay, and so skeletal muscles especially are obviously moving the skeleton here. So we have skeletal movement. But also cardiac muscle is going to be moving blood through the circulatory system. And then smooth muscle is going to be moving food through the digestive system. So here we have movement of different types. Also, maintaining posture is another job of the muscular system, of muscles in general. So either a standing or sitting upright position here is maintained by the muscles. A 4th function of the muscular system is to stabilize joints, especially of joints that are not really tightly articulated together. So here we see the shoulder, and so the muscles of the example here, the muscles of the rotator cuff are the ones that will help stabilize that shoulder joint there. Right? So stabilizing joints. And then lastly is temperature regulation, right? Generating body heat, so temperature regulation. And about 40% of the mass of your body is skeletal muscle. And so when those skeletal muscle cells are contracting, they are generating lots and lots of heat because they are using ATP, and breaking down ATP releases heat energy. So those are the 4 functions of skeletal muscles, or the muscular system in general. So it might be a good idea to pause and review those. So comparing the 3 different tissues, 3 different muscle types. First we have skeletal, and then cardiac, and smooth muscle. So the majority of the chapter is going to focus here on skeletal muscles, and we see that where they are located, obviously. So you can read through this table here, but we're going to look at body location and then the cell shape and appearance. So we spend a lot of time looking, of course, here at the cell shape and the appearance, because this is where most of the function is going to take place, right, at the cellular level. So skeletal muscles under the microscope appear striated or striped, and you can see those striations or stripes here, this alternating light and dark banding pattern here. You see the striations appear in the cardiac muscle as well, but you don't see the striations here in the smooth muscle. Okay. Another similarity and difference here is that the cells of skeletal muscle are known as fibers because they are long and fibrous, and the cells of the smooth muscle are also called fibers. The cells of the cardiac muscle tissue, though, are just called cardiac muscle cells. They're not long cylinders, they're not long fibers. In fact, the cells themselves are branching, and so here we see this kind of branching pattern to them. And there's another branch of a cell right there. So these are just the cardiac muscle cells. Incidentally, before we move on here, just a couple of prefixes or roots to know that you're talking about muscle. One is sarco, which means flesh, and so the flesh of the body. Again, 40% of the mass is muscle there. So sarco is flesh, and then myo or mice, these all MYS, these also just mean muscle, right? So in a lot of terms here, we're going to be talking about sarco or myo something or mycelium or something like that. Okay, so a good idea to kind of keep those in the back of your head or in the front of your head, as the case may be. So histologically, going back again here to these, you're going to see this is the skeletal muscle. Right, so looking for the striations. And if the cells appear to be branching, then they are going to be cardiac cells. The smooth muscle cells are sometimes the most difficult to tell. Sometimes that these also look like tendons under the microscope. So that's a tough call sometimes on those. But I'll try to give you ones that are obviously going to be either the tendon or obviously the smooth muscle there. Okay. Some other differences between skeletal, cardiac, and smooth muscle in general. How the contraction is regulated. Here, in skeletal muscle, it's a voluntary via the nervous system. So here we have a neuron coming down, and we're going to have this NMJ, or the neuromuscular junction. So the neuromuscular junction is going to be a synapse. And so we're going to look at that in detail in another screencast here. But there's a synapse between the neuron and the muscle. In the cardiac muscle, again, we have involuntary here, and this is controlled by a vagus nerve as well as other nerves involuntarily. And the heart also has an internal pacemaker, that SA node that we discussed in another screencast. So also nervous system, other nervous system controls as well as hormones. So the heart is under involuntary and hormonal responses, as well as a self-contained pacemaker depolarizing set of cells there in the SA node. Finally, smooth muscle, involuntary nervous system controls. Here again we see neurons, and so we're going to see a synapse right there. But also hormones, we'll be able to control that, different various chemicals in terms of ions and things, and then the amount of stretch is there again. So for example, the urinary bladder, when that is full, those stretch receptors there are going to help actually to contract that smooth muscle. Right? The speed of the contractions varies. In skeletal muscle, it's slow to fast, relatively slow, and then very slow. So the fastest is skeletal. Okay. And then rhythmic contractions, no here in skeletal muscle because it is voluntary, and the involuntary nature here, the yes, is going to be true for cardiac and smooth muscle. So good idea there to maybe pause now and sort of go back and review these different characteristics of skeletal, cardiac, and smooth muscle. So again, you should be kind of looking for how they are similar and versus how they are different. It might be a good graphic organizer to make there for yourself. Okay. So let's take a look at just a quick survey of the three different types. We're going to spend a lot more time looking at skeletal muscle, so I'll go over this relatively quickly here. Skeletal muscle, of course, is attached to bones, and here we see the muscle fiber, again, is one muscle cell. So the skeletal muscle itself, this organ, is made up of lots and lots of these individual muscle fibers. So that's a cell, that's a cell, 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 cell. So all these cells here, these muscle fibers, run the length of the muscle. Right? So these are why they are long, rather referred to as fibers here, is because the cells are these long columnar type things. And again, here we see them striated, and we can see that this is the microscope here. So here is this is one cell, and this would continue in this direction and continue out this direction, and the striations appear here as you see them. I want to point out the nucleus here. These nuclei are peripheral, and that means that they are off to the side, right? So notice that there's no big nucleus taking up any room right here in the middle of this cell. So you don't have a nucleus there. The nucleus is peripheral, and there are multiple of them, right? And so in one muscle cell, you have many, many nuclei. There's a nucleus and there's a nucleus. So skeletal muscle fibers are sometimes also described as being multi-nucleated. Okay. Cardiac muscle. We looked at cardiac muscle in the cardiovascular system in those screencasts. The arrangement of those fibers, or the cells rather, sort of is in this figure 8 pattern, right? And so these are the muscle cells of the right ventricle. You have these little skeleton fibrous rings here that hold the valves in place, right? And so if you were to zoom in now on these cells, you would see that they are roughly cylindrical, but they are branching cylinders, right? So all of this is the border of one cell, and it continues off this way, and then it comes down here. This is still all one of those cells. This is all a cell that I'm outlining here, and it continues off this way, and then this would be it. So all of this is one cardiac muscle cell. So they're not long cylinders. We don't refer to them as fibers, therefore. An interesting thing that's another thing here is that it has a nucleus of the cardiac cell, and that tends to be centrally located, not peripherally located. And we see then very clearly here these intercalated discs. And so an intercalated disc is the place where the cardiac muscle cells are attached very tightly together via these tight junctions, impermeable junctions. So these are proteins that are sort of anchoring these cells together, these cardiac muscle cells together. And so when one cardiac muscle cell contracts, is signaled to contract, an action potential can be spread across them very, very rapidly from one cell to the next. And as one contracts, it's going to pull on the others, and you get a very tight contraction network. So these cells that are bound together act like one big long cell because they are held together very tightly by these tight junctions, which are in most descriptions called intercalated discs. Lastly, smooth muscle tissue. We've looked at smooth muscle in the screencast dealing with the digestive system. So here we see a cross-section through the digestive system, and we see this this double layer here of the smooth muscle cells. And so these smooth muscle cells are also referred to as fibers, and they're running in two different planes. This plane and then this plane is coming out at you. So you have this circular plane and then the longitudinal plane there. So the longitudinal layer cross-section, the cells are going to look relatively round, and then they also have one nucleus kind of centrally located. Here, there's no nucleus in the cell because you took a cross-section through the cell where you did not, where the nucleus was not. So for instance, up here, if I made the section this way, if this was the plane of section, notice I caught this cell, I caught the nucleus of this cell, but there's no nucleus in this cell at that point of the plane of section there. So that's why you see no nucleus in some of these cells here. So the smooth muscle cells, again, sometimes look like tendons, but one way that you can tell them apart is that that you do see nuclei here in these cells. And again, a tendon is going to be mostly collagen fibers there, and so you're not going to see nuclei stained. That might be a tip-off there. Okay? And these smooth muscle cells tend to be tapered on the end, right? And so they have this sort of appearance. So they're not cylinders like a skeletal muscle would be, and they're not those branching type cells that the cardiac muscle would be. So this shape is called fusiform, and so they have this fusiform shape where they're tapered at this end, and then I didn't draw it very well, and then they're tapered at that end as well, and then they're going to have just a nucleus in the middle of them there. And the fibers that are the cytoskeleton cytoskeleton microfilaments here are connected in different planes of section. When we talk about skeletal muscle contraction, I'll bring this slide back, and this will make a little bit more sense. But here's the smooth muscle cell relaxed, and then here it is contracted. And so it's just going to get shorter in many different planes, sort of at one time. Whereas a skeletal muscle cell that is a long cylinder like this is going to get shorter just in this plane. Right? And it's not going to shrink down in size in this plane at all. It's going to stay that width the entire time, whereas the smooth muscle will change its size because of the nature of the cytoskeleton elements that are inside there. Okay, so a lot of information there, again, about the four functions, and then a lot about the characteristics of the three types of muscle tissue. And the following slides I'm not going to talk about, but you can look at on the PowerPoint. Will deal with the histological preparations of those three types, look at them under the microscope. So I would encourage you to take a look at those, and also the slides that we have available in the back of the room, and being able to identify these and describe them. Okay, thanks a lot for listening, and bring your questions to class.